So that decade really starts in the late 60s to 1980 was a marvelous time for cell biology. I mean, this work came out in that time was a revolution for understanding how synaptic transmission works. There was just an era of great cell biology. So why do you think that happened, and why did it end?
Well, I don't think it did end.
It didn't end.
It's the shift.
Are you kidding?
It has
It's just
shifted.
starting up again.
The focus is, molecular biology sort of swept away.
Oh yeah, I mean there was a period of time in the summer course, and there you get these guys from Caltech, and they say, well, in fact, we had a student, and she got up and she said, I want to let everybody know that electron microscopy is a dying field, and so we went and we made a plot of what a dying field would look like. We put it on the door, a dying electromagnetic field. Remember that?
No.
I just showed this thing, and all of a sudden the curve went down. We had a dying field. She was from Keith Porter's lab too.
Really?
The founder of modern electron microscopy, but they've become disillusioned. Yeah, so there was a period of time when people thought they were going to understand everything by understanding genes, and we've understood a lot from genes, but you don't understand everything from genes. Now, finally, the technical side of electron microscopes is now being addressed again, and new kinds of electron microscopes are appearing, and the challenges on people like John and me, people who actually prepare stuff for the electron microscopes to take advantage of these new kind of optical things that these microscopes can use, and that's going to happen big time. And we're going to suddenly understand there's going to be a whole new understanding of cells, not at the level of membranes running around, but of the individual protein molecules running around interacting with each other.
Back up to what Tom was talking about, the death of electron microscopy, that period when it was, its imminent death seemed unavoidable. There are a couple of other factors that I'd like to bring up. One is that was a period when light microscopy took off, when it got started. And light microscopy was providing two incredibly fundamental things that EM did not provide. Number one, the dynamics of cells, the actual movements of all of these things. Mind you, light microscopy is 1/1000th the resolution, that far away, but it was a whole process displayed for you in a living cell. Second, the light microscopy was allowing you to begin to identify which protein was doing it, because new technologies of fluorescent proteins and so on were coming in that could be examined by light microscopy. So it was not that electron microscopy died, it was eclipsed. It was eclipsed by light microscopy and the huge potential of it.
You went through a light microscopy phase.
I fell in love with it myself and lost myself in it. But
Never did.
The critical thing that we've always been up against, and we are still up against, and no matter how fancy the electron microscopes get or how perfect our means of viewing is, we will never get around the fact that the electron microscope gives you a static image, a still, a snapshot of something, one instant in time. For each picture that you take, each preparation that you make is one instant in time. And you have to back extrapolate to what was happening in life. You have to say, well, I timed it perfectly. I have 5 milliseconds and 10 milliseconds and 20 milliseconds, so I can say it did this and that and that. But that's an interpretation in itself, open to questions if you've got competitors in your field. It's always that extrapolation event. It's an interpretation, and that made people shy off and led to the demise of which we're now trying to recover. Always will be trying to recover.
I mean, if you were a young student in the course and you came in and you had a choice of spending a week doing meticulous preparation to get something ready for the electron microscope to get one picture of it, or you could take some genes and create some beautiful colors that would run up and down inside your cell, and you could sit there all afternoon looking at them, which would you do?
got me. I'd sit here and look at this. That's one picture.
Yeah.
So what is the future of electron microscopy then? What recommends it to students?
Well, you asked two questions there. The future and what recommends it to students. It might be that there's little to recommend it to students. That's what we're really facing.
Yeah.
How many people are going into this field? It's heartbreaking to see how few people are being trained, even in these labs that are totally cutting edge. And the whole culture of electron microscopy is in dire danger.
So still, you haven't answered why they should be trained. What are they going to see that you can't see in a light electron microscope?
Everything. I mean, you're very limited. The idea people got all excited about high resolution light microscopy, super resolution. Super resolution, that's about a 50 or 100% improvement maybe, but you go from seeing something that's 200 nanometers to something that's 50 nanometers. But we see stuff all the time that's 1 nanometer, 2 nanometers. I mean, the gap between resolution and the light microscope and the electron microscope is still huge.
So,
and my goal in my current work is I want to understand how individual proteins fit together to make protein motors. Now you can examine that with light microscopy with some fluorescent dyes and watch what happens. You can make some inferences, but you don't really know until you actually get the images and reconstruct the proteins.
I've just listened to Tom give his lectures to this year's students at the Marine Biology Laboratory, and I'm impressed again by what a purist he is. I mean, as he's saying, and it's apparent in every word he says to students, he believes, and I totally agree with him, that it's at the EM resolution, the level of the electron microscope, that we're going to understand cell function, organelle function, everything about the cell, because it's at that level that you can actually see molecules. And the whole name of the game is seeing molecules interacting with each other. To understand this machine called life, we have to see the individual molecules interacting with each other. Light microscopy gives you these wonderful clues that, they're dancing together, they're going over here, they're going over there. But to see how that machine works, you have to have the resolution of this instrument. And it's a simple fact. It's amazing that we have the instrument. It's great that we've had it for 50 years, 60 years almost, but it's got a niche in cell biology and in medical science that is irreplaceable. Never will be.